Hello guys, welcome back to this channel. Today in this video, we'll be talking about hybrid dysgenesis in Drosophila melanogaster. Before talking about hybrid dysgenesis, we must first know about transposable elements or transposons they are called, but the appropriate term is transposable elements. There are two types of transposable elements. One is the retrotransposons and the other is DNA transposons. These DNA transposons are also called simply as transposons. Okay. It is due to a type of DNA transposon which is present in Drosophila melanogaster that this hybrid dysgenesis occurs, and that transposable element is called P element. Okay, and this P element is a transposable element which causes a different kind of mutations, chromosomal aggregations in the Drosophila melanogaster oocyte, and thus causes sterility in the hybrid. That's why this is also called as hybrid dysgenesis, which means the hybrid produced from crossing two different strains is sterile because of the dysgenesis by this P element transposon. So how this P element causes this hybrid dysgenesis in Drosophila? For that, we must understand that there are two types of strains, two strains in Drosophila melanogaster. One is called P cytotype and the other is called M cytotype. Okay. In case of P cytotype, there is presence of P elementus. Okay. And there is also presence of P repressors. Pre-repressors are those proteins which repress the transposition of P elements in them. Okay. So P repressors and P elements both are there, but these P elements are passive. Okay. In case of sorry, in case of M cytotype, there is no repressor. There is no repressor found. Okay. And there is also no P elements. Okay. And whenever we cross these two cytotypes with each other, we may either get a fertile hybrid or we may get a sterile or hybrid dysgenesis produced hybrid, which is sterile. Okay. So let's look into that. Okay. So here is the first case where if you take a P cytotype female and you cross it with an M cytotype male. Okay. Here in case of female, there is presence of, in case of P type female, there is presence of P repressors. P repressors in the cytoplasm, and these P repressors can be inherited by cytoplasm as well. So in case of male, there is no P repressors, okay, and no P elements as well. So what happens here? The hybrid producer will be having these P repressors in the cytoplasm, okay, and hence the P element is again inherited from the mother, will not be able to move. So this hybrid here, so this PM hybrid here will be fertile, will not be sterile, we should say. Okay, hybrid is fertile, and when you kind of switch the two sexes, that means you go for a reciprocal cross. So in case of a reciprocal cross here, you take a P cytotype as male, okay, and you take a PM cytotype as female. Okay, so in case of this cross, the P repressors are present in male, and so are the P element is present there. Okay, and there are no repressors here in case of male. No, sorry, in case of female, there are no repressors and no P elements. Okay, and when a hybrid is produced from this very cross, it will inherit only the DNA from male. Okay, and it will inherit this DNA plus cytoplasm from the female. Okay, but there will be no presence of P repressors, and the P element is coming from the father. Okay, will undergo a hell of a transposition and will cause dysgenesis. Okay, so we can write no repressors, no repressors in hybrid, but P elements are present. But P elements are present. As a result, there will be transposition, and hence sterility in the hybrid. Sterility of hybrid. Okay. So I hope you got this. Still, I will try to repeat it again, and it will make much more sense. So we said that hybrid dysgenesis occurs because of a transposable element, okay, which is a transposon, DNA transposon named as P element. Okay, we said based on the activity of this P element, we have a P cytotype and M cytotype. In case of P cytotype, there is presence of P elementers and P repressors, and these P repressors then suppress the activity of the transposition of P elementers. And in case of M cytotype, there is no repressor and no P elements. Okay, and when we cross these two strains with each other, as you can see here, uh, the in the first case, when you take P cytotype as the female and M cytotype as the male, you get a hybrid which is fertile, because this hybrid will inherit P repressors and P elementals from the mother. While as in case of the reciprocal cross, where you take P cytotype as the male and M cytotype as the female, there will be no repressors in female parent, and thus no repressors in the hybrid as well. But this hybrid will inherit P elements from the male. Okay, and these P elements will undergo transposition and will cause sterility of the hybrid and will cause hybrid degenesis. Okay, so that's it from today's video. I hope you got the gist of this idea, and if you did, kindly do subscribe, like, and share this video. I will see you in the next video. Till then, take care. Bye bye.